So our next speaker is Dr. Giuseppe Marrazzo. Giuseppe has a PhD in cognitive neuroscience and works on emotion.
Actually, I'm a PhD student, not yet.
Oh, I'm sorry. Giuseppe is a PhD student in cognitive neuroscience and works on emotion and body perception. He is working in the lab of Professor Beatrice de Gelder at Maastricht University. And today, Giuseppe is going to talk about the dynamics of body category and emotion processing in high-level visual prefrontal and parietal cortex.
So can you see my screen?
Yeah.
Okay, thank you, Elaine, for the introduction. So I'm Giuseppe Marrazzo, PhD student at Maastricht University. I work with Professor Beatrice de Gelder in the brain and emotion lab. And today I'm going to talk about the task dynamics of body category and emotional representation in high-level visual prefrontal and parietal cortex. So different studies have provided detailed understanding of how a familiar object like the human body is processed. And we also know that the brain encodes stimulus information in a high-dimensional representational space. This encoding process appears to be dynamic, relatively task-sensitive, and it also may be at service of different and complex behavioral goals. What is actually less clear nowadays is to what extent specific body attributes like the emotion, for instance, might influence the activity of these body areas, and also whether the task performed may impact on the representation of this object category. So, in order to clarify this situation, in the present study, we measured the representational dynamics of explicit and implicit body expression perception, and we investigated whether the type of task influences the representation of bodies and body expression inside and outside the body selective category areas. We used fMRI data of 20 participants scanned while they viewed still images of angry and happy body postures of Black, African, and white Caucasian ethnicity. Our experiment comprised 2 categorization tasks over 4 separate runs. Each run consisted of a presentation of, and let me point it out just for clearness. So our experiment comprised 2 categorization tasks over 4 separate runs. Each run consisted of a presentation of an emotion block, as you can see here, and a shape block. During the emotion block, the participants judged whether the emotion expressed was anger or happiness, here at the bottom. And during the shape block, participants judged whether the shape which was superimposed on the body was a circle or a square. For what concerns the analysis, to analyze the brain responses, we used the Neuromatch GLM, searchlight multivoxel pattern analysis, and representational similarity analysis. On the left side, we have the results of the Neuromatch GLM when contrasting the activity within the 2 tasks. The color map here indicates the region where higher in red or lower in blue activation was found basically for the emotion recognition task versus the shape recognition task. On the right side, instead, we have the above chance classification accuracies produced by the classifier when trained to discriminate between the 2 tasks. The color map here indicates the t-value of the group pair sample t-test performed against chance level. It is worth noting that ventrolateral prefrontal cortex, IPL, and EBA are among the areas where there is max the difference between the 2 tasks, and also ventrolateral prefrontal cortex and IPL appear to be more active in the explicit task and in the implicit task, respectively. These ROIs are instead the results of the classification accuracies tested against 0 produced by the classifier threshold at the Q of FTR 0.01. On the left side of the panels, we show the representational dissimilarity matrices computed with 1 minus Pearson's correlation distance between the different conditions, which here are labeled in red for the emotion explicit task and in blue for the shape implicit task. On the right side of the panels, we find the group averaged ROI beta values plus the standard error. It is interesting to notice that the representational dissimilarity matrices for ventrolateral prefrontal cortex and EBA present a pattern of similarities within the explicit condition. From this, we see that the difference between explicit and implicit body expression processing can be decoded in these two areas, although no emotional specificity there is. On the same page, we see that there is a higher activation for the explicit task in the same two areas, while on the opposite, we have a higher activation for the implicit condition, so the implicit task for IPL. So to wrap up, in this present study, we measured the representational dynamics of explicit and implicit body expression, and we identified the brain areas that are critical for the distinction between these two tasks. Our results indicate that the task affects body category representation, and in the specific EBA may be active in response to explicit body attribute recognition. Ventrolateral prefrontal cortex may also itself play a role in this categorization because it also codes for body category when the task demands it. And as take-home message, we can say that the combination of higher activation in EBA and ventrolateral prefrontal cortex and respectively lower activation in IPL suggests a connection between these three areas, with the ventrolateral prefrontal cortex possibly influencing EBA positively and IPL negatively. Moreover, we can say that the task performed changes the object category representation, and last but not least, depending on whether the emotion recognition is explicit or implicit, different brain areas are involved in the process. So thank you very much. If you want to know more, have a look at the paper.
Thanks very much, Giuseppe, for that's a really interesting talk, a really nice demonstration of how higher level neural representations are context dependent. I was wondering how much of this would you think is specific to emotion processing, or would you find, do you think, similar findings if you had some other body expression discrimination task?
For bodies, it has been shown by Wager and Bugatus and Bracci that there is task sensitivity in frontoparietal areas. The most interesting thing is that this task effect is usually confined in frontoparietal areas. So there is no evidence of a task effect in the literature or in a high-level visual cortex, or in more specifically ventrotemporal and ventrotemporal cortex. Let's say that EBA, so specific body areas, didn't show a task effect, let's say, so far. So there is for sure a task effect which is not related directly to emotion, but only the body attributes, let's say, and has been shown by previous studies. And here I also put the references about it.
And I was wondering about whether there was a difference in difficulty across the two tasks, or whether that was
there is I didn't report the behavioral analysis because yeah, I mean 5 minutes it's not a lot, but yeah, in general there is a significant difference, but I don't think it's a substantial difference. For instance, for the accuracy values between the two tasks, we have 96% accuracies for the implicit task and 90%, I don't remember exactly the digits, but 90% for the explicit task. So this difference appears to be statistically significant, but at the end of the day is not, let's say, substantial. So the tasks were designed to be very similar in order, I mean, really to rule out the possible I mean, differences in the task difficulties.
Did you look at all at whether the difficulty might change over time? Was there repeated exposure to the same stimuli?
I mean, I didn't get it, sorry.
I don't know. I was just thinking about whether if you were repeatedly exposing them to the same images, would people kind of be
sort
find
of
it easier?
adaptation, you mean sort of
Yeah.
adaptation? Yeah, I mean, this we didn't actually try it because, yeah, of course, it's always the time that you have for performing the experiment is limited, and yeah, so we didn't actually check for adaptation. We do have a certain amount of repetition of each unique stimuli, of course. I think it's 5 repetition of each unique stimuli, and this didn't rule out, let's say, didn't influence too much, let's say, the behavioral results.
Okay. Well, we had a third talk lined up. If nobody has any more questions for Giuseppe, I'm not sure if our third speaker is here.